Okay, so we're going to start Chapter 8, which covers the somatic senses as well as the special senses. So sensations are the conscious and subconscious awareness of changes in environmental stimuli. Somatic sensations include things like tactile or touch, pain, temperature, and proprioception, which is our body or joint position, whether or not something's bent or whether you're sitting or standing. Visceral sensations include things that are occurring on the insides of our body. Pain, as well as blood pressure, chemical levels like pH or oxygen and carbon dioxide concentrations would be included in that. Specialized senses include those senses that we have special sense receptors for: sight, smell, hearing, balance, taste. All of these occur within our central nervous system. They're detected by nerve receptors at a particular location in the body, and the nerve receptors are specific to a specific sensation. That was hard to say. Somatic and visceral sensation receptors are spread widely throughout the body. You need to know if anything on your skin is hurting you or burning you, right? You need to know if there's pain going on the inside of your body, no matter where it is. So all over the body are those types of receptors. Our specialized sensation receptors are localized within the sense organs, like our nose, our ears, our mouth, our eyes. Stimulation of any of those receptors is going to initiate that signal or action potential within that dendrite, and then that action potential can then be relayed to the central nervous system, where information processing takes place, and then a motor command or some type of body response can then be carried out. So this diagram shows you some examples of somatic sensations. Things like tactile sensations include touch, as well as pressure and vibration. Thermal sensations like heat or coldness. Nociception is pain. Proprioception is joint muscle position, as well as movement of the heads and limbs. These receptors are all over within our joints and muscles, within the skin, as well as within the organs. Our special senses are within the nose, eyes, ears, and mouth. The visceral senses are going to monitor conditions within the organs, things like pain, level of chemicals, as well as blood pressure. Our special senses, as I mentioned, sight, smell, hearing, and balance, which are all within the ear, as well as taste. Adaptation is the term used that describes the decrease in strength of a sensation during a prolonged stimulus. For example, if you're sitting in a chair right now, maybe when you first sat on the chair you realized that it was cold, or you realized that it was warm. Now you don't realize it anymore because you've adapted. Or maybe you like to wear uncomfortable shoes, and as soon as you put those shoes on, you're like, oh, this is why I hate wearing these shoes, but they look cute anyway, right? After an hour or so, you may, hopefully, forget that they're as painful as they actually are. Your body has adapted. Maybe your dogs really stink, and as soon as you smell them for the first time in a week after being away, you realize, whoa, these dogs really stink. But when you're living with them on a day-to-day basis, you forget that they stink. That's because you have adapted to their sense of odor. Somatic sensations occur on our skin. Our dermis, remember the dermis, just deep to the epidermis, that middle layer there. It contains special tactile receptors, which I mentioned in the chapter on skin, chapter 4, I believe. Those tactile receptors are encapsulated nerve endings, so they're basically the axon terminals of various sensory fibers. They're enclosed in a connective tissue capsule. They include receptors like Meissner's corpuscles, Merkel's disc, Ruffini corpuscles, hair root plexus, and Pacinian corpuscles. There's also free nerve endings like nociceptors and temperature or thermoreceptors, and some touch receptors are included in the free nerve endings. Here you can see the structure of some of these somatic sensations. Here you see the hair. So let me get my mouse together here. Here you see the hair, and there's a special nerve fiber wrapped around the follicle. That's the root hair plexus. There's a Pacinian corpuscle. This guy is going to respond to deep pressure. Think of the Pacific Ocean being very deep. Pacinian corpuscle, Pacific Ocean, deep pressure. The root hair plexus is going to respond to movements of the hair. So as that hair blows or moves, it's going to stimulate this root hair plexus going around the follicle. The Ruffini corpuscles are going to sense stretching, and I think of stretching, I think of ruffles. When things are stretched, they're not ruffly. anymore, and that Ruffini corpuscle looks like the word ruffle, so stretching deep in the skin. You have tactile discs or Merkel's discs, which are actually within the epidermis. They're almost fused with that basal layer, so they're very near to the surface for fine touch. And then the Meissner's corpuscles, which are within the dermis, responding to fine touch as well. You then have free nerve endings like these nociceptors here, which end in the epidermis to sense burning sensations or temperature, etc. So, to review them again, the Meissner's corpuscles are touched near the skin surface. The root hair plexus is for movement of hair, such as wind, right? The tactile disc is touched near the skin surface as well. The big difference between the Meissner's corpuscles and the Merkel's disc is the location. Those Merkel's discs are actually within the epidermis or within the basal layer of the epidermis, whereas the Meissner's corpuscles are within the dermis. The Ruffini corpuscles, think of that word ruffled, stretching, going back to that word ruffled, stretching within the deep layers of skin. Whoops, went back the wrong way. Pressure receptors include our Pacinian corpuscles. These are found throughout the body. They're sensing deep pressure as well as high frequency vibrations. Here you can see them, the Pacinian corpuscles. The nociceptors are those free nerve endings. They sense pain. They do not adapt. They can be fast acute pain receptors or type A receptors, or they can be slow chronic pain receptors or type C fibers. The fast acute pain receptors, these guys have that sharp prickling pain that they're going to sense, like touching something sharp. I think of the other day, I stepped on a piece of glass. It was my fast pain receptors that felt it in an instant. That way that glass did not get into my foot that deep. A slow chronic pain receptor is that itching, or rather aching or burning or throbbing pain that you get from time to time. Maybe you have a bad back and your back is always hurting you. It's those nociceptors that are slow and chronic that are constantly being told that there's pain. Pain can also be misinterpreted. Pain from internal and visceral organs can be felt in an area that is not actually associated with that structure. That's called referred pain. You may know this already and you don't even realize it. People that are having a heart attack, what part of their body hurts them? Their left arm, you may recall. And that's because there's referred pain. And that has to do with how those sensory neurons are synapsing within the spinal cord. They're triggering other interneurons to make it feel like it's in a different part of your body. People that are having a gallbladder attack. Gallbladder is just underneath your right rib cage. People that are having a gallbladder attack frequently feel it in their shoulder. Very strange, but it's because of the way the nerves are interacting in the spinal cord. Here you can see what I was describing here with the heart attack pain down the left arm. The liver and gallbladder would be on the right side shoulder area. Different areas of your abdominal pelvic cavity corresponding to different types of pain, as well as parts of your back can respond to those types of pain too. Proprioceptors are found within the skeletal muscles. They're also found within the tendons and within the joint capsules. They adapt very slowly and not much at all. Your body's always aware of where it is in space. Your brain's receiving constant information about head and limb position as well as movements. And that's important. It's important for your body to know where it's important for your brain, I should say, to know where your body is. That way it knows how to respond or react if a threatening situation came about. So where are these somatic sensations processed? Well, if you recall, all of that sensory information comes in through that dorsal root ganglion, goes up that posterior column pathway through the spinal cord, synapsing with the upper, or rather second neurons within the medulla oblongata. It's going to cross over to the other side of the body for that lateralization. It's going to synapse within the thalamus. The thalamus is always relaying all of our sensory information. And then it's going to go up to that primary somatosensory area of our parietal lobe. So there's two main pathways that it could follow. There's one pathway that's for touch, pressure, and vibration. And there's another pathway for cold, pain, warmth, and itch. There's also proprioceptors that are going to pass this cerebellum to aid with movement coordination. So this path may vary depending on exactly which sensation is coming in. Let's talk about our special senses now. We have two special senses which require chemoreceptors or special receptors for chemicals. That's olfaction, our sense of smell, and gustation, our sense of taste. The nose and mouth are connected, so smell and taste are also connected. And you may recall this as a child. If you were ever asked to eat something you didn't like, or who knows, maybe you still do this, you pinch your nose before you eat that broccoli or those lima beans or whatever that you may not like. Let's talk about our sense of smell more specifically. There's 10 to 100 million olfactory receptors in the nose, so it's very, very receptive. It's within a structure called the olfactory epithelium. That's basically the inner lining of our nose. It's surrounded by special supporting cells and basal cells that can actually regenerate. They bind and recognize around 10,000 different chemical molecules called odorants. Odorants are then going to bind to that olfactory epithelium, initiate an action potential, and that action potential is going to travel back through cranial nerve 1, which you may recall is the olfactory nerve. It's then going to go back to the olfactory areas of the brain within the temporal and frontal lobes, within the limbic system, which is where all of our emotion is, as well as the hypothalamus, which controls our body's homeostasis. The limbic system stimulation can evoke strong emotional responses and memories, which is why maybe if you smell some cheap cologne one day, you're reminded of your 6th grade boyfriend or something like that. Typically, our sense of olfaction has a low threshold for stimulation. In other words, it doesn't take much to make our sense of smell sense something, but they adapt very rapidly to odors, like I was mentioning with the stinky dog. You are away for a while and you may not realize how stinky your dog is until you come home, and then you realize, whoa, he really needs a bath. But if you're with him over the weekend, you may forget how stinky he actually is. As I said that my dogs gave me the evil eye, they must know I'm talking about them. So here's the structure of the olfactory epithelium. Within the brain case itself, you have the bone, a special part of the ethmoid bone called the cribriform plate. The cribriform plate holds in place a structure called the olfactory bulb. The olfactory bulb is made of lots and lots of sensory neurons, which synapse with these neurons or axons of our olfactory receptors within the olfactory epithelium. So this right here, this soft blue lining, is actually the mucus lining the inside of your nose. So air passes over this blue lining. You can see these little particles, these odorants, get trapped in the mucus, bind to the receptors. The receptors are then going to communicate to our olfactory bulb and our olfactory neuron, or nerve rather, which sends the signal back to the olfactory cortex. The mucus is necessary. In fact, in order for our sense of smell to work, as well as our sense of taste to work, the chemicals have to be dissolved. If you completely dry out your nose, you will not be able to smell anything. If you completely dry out your mouth, you will not be able to taste anything. Speaking of taste, let's talk about our sense of gustation. Gustatory receptors are also known as taste buds. They bind to tastins, or chemicals that are within our food. There's 5 primary tastes: sour, sweet, bitter, salty, and umami, which is a meaty or beef broth type of taste. In the brain, the gustatory center combines with those 5 tastes and the associated smells to allow us to detect the flavor. These tastins dissolve in the saliva, just like the odorants dissolve in the mucus. The taste enters the pores, contacts the special receptors, the gustatory receptors on our taste buds, and the gustatory receptors transmit that information via the 3 cranial nerves that are responsible for our tongue: cranial nerve 7, cranial nerve 9, and cranial nerve 10. That's the facial nerve, the glossopharyngeal nerve, and the vagus nerve. Back to the primary gustatory area, which is in the parietal lobe, also with the limbic system to get some emotion in there. That's why a lot of us are emotional eaters, and it feels good to eat. And the hypothalamus controlling our endocrine system for all of those digestive secretions. The taste and information relayed to the limbic system, as I said, can also evoke strong memories. Maybe eating some type of special hard candy might remind you of your grandparents, for example. Adaptation is very rapid with gustation as well, which is why maybe once you dig into that bag of salt and vinegar potato chips, that first bite, it kind of gets you because it's really salty and really vinegary. However, after you're halfway through the bag, you forget how salty and how vinegary those chips really are. That's because the adaptation is very quick. Taste perception of different tastes come from different patterns of activation of those taste buds, as well as activation of the olfactory receptors. So it's a very complex sense. Here you can see a diagram of the tongue, and it's also showing you where this various lingual papillae are. Papillae are the what we call taste buds, but they're not actually taste buds. They're just the different regions of our tongue. They have different purposes. The majority of the papillae that are associated with taste buds are towards the back of our tongue as well as to the sides. If you put something only on the very front of the tongue and your tongue is dry, so whatever chemicals are on it aren't allowed to diffuse to other parts of your tongue, you probably won't be able to taste it, because the majority of the taste buds that are on the very tip of your tongue don't have any taste buds associated with them. You can see how the taste buds themselves have little pits on them, which are, or I should say the papillae themselves have little pits on them where the taste bud receptors are, and as we eat the food and it dissolves in our saliva, it goes into these pits and is absorbed by these taste bud receptors, which then communicate through a gustatory sensory neuron. Let's move on to the next sense, which is vision. Let's talk about some protective structures of the eye. Again, in lab, you may review some of these structures, or you, excuse me, or you may even have to know some of these structures for a practical. The protective structures of the eye include the eyebrows and eyelashes, which help prevent the foreign objects from coming into the eye. Also, eyelids to cover and protect the eye, as well as to protect it from drying out or desiccating. The lacrimal apparatus, which is secreting tears and drains fluid to moisten the eyes. The lacrimal fluid itself, which is our tears, containing salts, mucus, as well as an antibacterial enzyme called lysozyme. And there's the lacrimal glands, which produce the tears. Here you can see them. Most people think of that little fleshy structure in the corner of their eye as their tear duct or tear gland, and that is in fact not correct. The tear glands or lacrimal glands are actually on the lateral side of your eye. They secrete through little ducts just underneath your eyelashes, and that lacrimal secretion or tears sweep across the surface of your eye, drain into these little canals called puncta or ducts, and they actually drain down your nose, which is why when you're crying, if you really are crying good, you get into that ugly cry, your nose also starts running as well, because it's draining right into your nose. You can see on this picture of the eye some of the accessory structures, the eyebrows, the eyelashes, and some of the other visible structures of the eye, including the pupil, that nice black circle in the middle. The iris is that colored muscle, and the conjunctiva, which is the outer lining of the eye. It's over a certain area of the eye called the sclera. So here you can see some of the extrinsic eye muscles or exterior eye muscles that help move our eye around. These are the muscles that control whether we look right or left or up or down. We just take a second to look at some general structures here. You can see some of the special muscles that move our eyelids. This would probably be our orbicularis oris or oculi muscle going around our eye. Rooted to our eye itself is a superior rectus muscle. This muscle is going to contract and allow our eye to look up. And then we have an inferior rectus muscle, which when contracted will allow our eye to look down. Notice our eye socket itself, or orbit, is surrounded with lots and lots of fat. That's there for protection and isolation. As we age, those subcutaneous fat deposits move to other areas of our body, and that makes our eyes look saggier. Let's talk about some of these internal eye structures while we're looking at this picture. Here you can see where our eyelids would open right here, would allow the light to come in, go through, go across the cornea, through the pupil next to the iris. This blue structure here would be the iris. Through the pupil, through the lens, through this posterior chamber or posterior segment of the eye, and then the light would reach the back of the eye, which is where the retina is. The retina is made of lots of neural tissue, which collects all of that light information and gathers it in those nerve axon bundles, leaving the eye as the optic nerve. Our eye has extrinsic eye muscles, as I pointed out on that last picture, the extrinsic and, rather, superior rectus and inferior rectus. But there's also intrinsic eye muscles, muscles that work within our eye. Our iris is an intrinsic eye muscle. It's controlled, if you remember, via the sympathetic and parasympathetic nervous system, the autonomic division of our nervous system. As our pupils constrict in bright light or under parasympathetic innervation, and our pupils dilate under sympathetic innervation or in very dim light to allow lots and lots of light into our eyes. There are layers of the eyeball called tunics. The outermost tunic is called the fibrous tunic. It's made of the cornea and the sclera. The vascular tunic contains the blood supply and secretes fluids for the eye. This is made of a structure called the choroid, as well as the iris, and another structure called the ciliary body. The neural tunic, or retina, is the nervous tissue layer, and this is where that light is conducted or changed into nerve impulses. There's also two chambers of the eye. The chambers of the eye are separated by the lens. The anterior cavity is filled with an aqueous humor. If you get a chance to dissect an eye in lab, you'll see that the aqueous humor is a very watery-like fluid. This aqueous humor helps to maintain the shape of the eyeball as well as to nourish the lens and the cornea. It has a composition similar to CSF. It drains through a small canal called the canal of Schlemm. When that canal gets blocked or is no longer functioning, the individual may develop a cloudy anterior cavity, in which case you would be diagnosed with glaucoma. Glaucoma is increased intraocular pressure, blocking off that canal of Schlemm and not allowing that anterior humor to drain would cause glaucoma. The posterior cavity, which is the area behind the lens, is filled with a jelly-like substance called vitreous body or vitreous humor. This prevents the eyeball from collapsing, so it gives our eye that three-dimensional shape. It also helps to hold the neural tissue of the retina in place. So there are several layers to that retina on the back of the eye. There's connecting ganglionic cells, there's interneurons and supporting cells, as well as photoreceptors which detect light. There's two different types of photoreceptors: rods, which detect shades of gray in dim light, and then the cones, which detect various colors in bright light. When it's dark out, or when you're meandering through the kitchen for a midnight snack and you don't turn the light on, your rods are active. When it's daylight, when you're looking at things in color, your cones are active. Here you can see a cross-section of the eye, and I'll walk through all of the parts with you. Let's start at the front where the light enters. The light enters and it's going to cross that cornea, which emits the light and allows it to bend. That light is going to pass through that anterior chamber or anterior cavity, which has the aqueous humor. It's going to pass through the hole within the iris called the pupil, this little blue area. It would be the circular muscle. We're just looking at it from a different angle. That's the iris. The pupil's the hole. So that light goes through the hole, goes through the lens. The lens is actually more like a soft grape than a hard marble, so the lens can kind of flex as well. The lens is held in place by these special suspensory ligaments, and those suspensory ligaments are going to fatten or flatten the lens to allow that light to go right back to that special area on the back of your retina called the fovea centralis. So as the light passes through the lens, it's then going to pass through that posterior chamber or posterior cavity, through that vitreous humor, and whatever you're focusing on. So if you're looking at the computer screen right now and you're looking, say you're looking right at this arrow tip right now, where my mouse is, the light coming from that arrow tip is bouncing through your cornea, through your lens, to the back of your retina, to the part called the fovea centralis. Everything else in the room that you're looking at that's not in fine focus, the things that you can see on the periphery, that's all the light that's coming to all of the other parts of your retina. Notice in this picture, you can also see the medial rectus on the medial side of the eye and the lateral rectus on the lateral side of the eye. You can also see the sclera, which is that thick fibrous layer. You can see that choroid, which holds in place the blood vessels and helps to absorb scattered light as well. All of our photoreceptors, rods and cones, the fibers of those neurons are gathered together in what's called the optic nerve, that leaves the eye at a location called the optic disc. The optic disc is also called the blind spot because you have no photoreceptors there. So if light were to come into your eye and go straight back to that blind spot, you actually would not be able to see whatever is in that pathway. This is the microscopic structure of the retina. Most students think that when light comes in, the light goes straight to the rods and cones, and the rods and cones collect the information and send it straight out to the optic nerve. That, in fact, is not the case. The light comes in from this direction. I wish I could draw on this, but I can't. The light comes in from this direction and passes past these ganglion cells, past the bipolar cells, past the rods and cones, and hits a pigmented layer. This pigmented layer is actually very, very, very dark brown, almost black in color. If you recall from your high school physics class, the color black absorbs all wavelengths of light. The color white reflects all wavelengths of light. Because this layer is pigmented so dark, it absorbs all wavelengths of light. When it does, it absorbs those light wavelengths, and it's going to trigger these rods and cones to send the information to the brain. The rods and cones interpret the information that was absorbed by the pigmented layer. They communicate that information with these special bipolar cells, which act as interneurons. Then these bipolar cells transmit the information to ganglion cells, whose fibers gather together and form the optic nerve. So let's talk about how an image is formed in the eye. An image is focused on the retina where the photoreceptors convert the light into action potentials. Refraction occurs as the light bends through different areas of the eye. Refraction is going to occur when the light comes into the eye at the cornea, when the light goes into the lens, and when the light leaves the ends. The majority of the refraction is determined by the contour of the cornea as well as the shape of the lens. The lens can be reshaped by the ciliary body, which can adjust to the refraction of the light. So let me back up just a minute so you can see what I'm talking about here. In this picture, you can see the lens, which again I mentioned is kind of like a soft grape. You can see the ciliary body, which holds these suspensory ligaments in place. The ciliary body will contract to fatten or flatten the lens, depending on how the light has to refract, to get that light to go straight back to that fovea centralis, as I mentioned. Ultimately, it's the shape of your cornea, or this outer layer, that's going to determine the major refraction, but some refinements can take place with the lens. Accommodation is the process of adjusting the shape of the lens for focusing. This changes, or this occurs, I should say, as you adjust your vision from far vision to close vision. If you're staring at your computer screen right now, look up across the room at something far away. Your eyes just accommodated. What had to happen was your ciliary body had to either contract or relax those suspensory ligaments, which are connected to your lens, to allow the light coming from that distant object to go back to your fovea centralis. If you now look back at your computer screen, accommodation had to happen again. Again, your lens had to either fatten or flatten to allow that refraction to get the light back to your fovea centralis. Rhodopsin is a pigment that's induced in the presence of light. It absorbs light and it undergoes a structural change to become an action potential. Rhodopsin pigment is going to accumulate in the presence of light so that when the lights go out, your eyes have an opportunity to see even in dim light. This is why when you turn out the light, if you were to turn out the light in your room and make it completely dark, at first you would take a few minutes to be able to see. At first it would be very, very, very dark. Then after a minute or so, you would slowly be able to make out different shapes in the room or where different pieces of furniture are in the room, just based upon a contrast between different shades of gray. That's because of that pigment rhodopsin. It works with your rods to allow you to see in the dark. This is showing you the idea of refraction. So the light before refraction is going at this angle, and then when light hits the water, it bends and is now going at this angle. You've seen this if you've ever looked into a fish tank. If you look at the sides of a fish tank, the fish look like they're in one position, but then if you take off the lid to the fish tank and look into it just through the water instead of through the glass, you'll see that the fish are in a different position. It's because of refraction. This is an image of the retina through the pupil of the eye. In lab, you may get a chance to use an ophthalmoscope to look into your lab partner's eye. In the back of the eye, you'll see lots of blood vessels. The blood vessels are radiating out from that central artery and vein coming through that optic disc at the back of the eye. This is where the blind spot is, so there's no photoreceptors there. This darker area is called the macula lutea, and the fovea centralis is the center of the macula lutea. That's your point of finest visual acuity. So again, this is where all of the light from whatever it is you're focusing on is going to in the back of your eye. This slide goes through some differences or maladies in lens shape and refraction. So let's talk about this example here. The light is refracted as it enters the eye through the cornea. See how it bends slightly as it comes through the cornea? This is when we're looking at something at a distance. The light then passes through the lens, and then that light carries the information back to the retina. The nerve impulse is connected back to the brain via the optic nerve, which is actually cranial nerve 2. I don't know why it calls it cranial nerve 1. That's a typo there. The photoreceptors within the retina transduce that light image into an action potential, which is conducted. If you're then going to look at something that's close, look at now the shape of the lens as opposed to this shape of the lens. Here we were looking at something far away. Here we were looking at something close. Refraction happens as it comes into the cornea, and then it's going to happen again when it goes through our lens. In this case, looking at something close is going to cause our lens to fatten to change the angle of refraction, ultimately getting that image to that fovea centralis at the back of our retina. When you have abnormal refractions of the eyeball, you may have a focal point that's either in front of the retina, or a focal point that's in back of the retina. An amyotropic eye is an eye that does not require lens correction, does not require glasses. So in other words, if you don't wear glasses, your lens and cornea are able to reflect the light correctly at the back of your retina. If you have a myopic eye, that means you are nearsighted, and what that means is where the lens and cornea refract the light is just in front of your retina, which means when that image actually gets to the back of your eye, to your retina, it's out of focus. If you have a hyperoptic eye, that means that your point of refraction is actually behind or posterior to the retina. These two conditions, myopia and hyperopia, can be corrected by different shaped lenses. If you're nearsighted, you're probably using a concave lens as part of your bifocals or glasses. This is allowing the light to come in, bend before it gets to the cornea, where it bends again, and then it's going to bend again when it hits the lens and get to that point, that sweet spot called the fovea centralis at the back of your eye. People that are farsighted are going to use a convex lens to get that point of focus back to the fovea centralis. Let's talk about the stimulation of the nerve impulse by light. As we mentioned, rods and cones are both sensitive to different impulses. In this case, we're looking at a rod being stimulated. So we'll start with step number 1. There's a change in shape causing the nerve impulse on the retina. Light comes in, and opsin, which is a protein, it's kind of the precursor to rhodopsin, becomes altered. That altered protein is going to dissociate from or it's going to dissociate itself, break into two parts. An enzyme is going to convert the retinol back into the original configuration, and retinol and opsin combined to create rhodopsin, this colored pigment. This colored pigment is what allows you to see light even when it is dark. Let's talk about the visual pathway from the eye to the brain. So again, the action potential has to go through the optic nerve, which is cranial nerve 2, to the visual cortex, which is all the way in the back of the brain at the occipital lobe. The impulses are integrated to form a representative image from the visual field of each eye. Some of this information is relayed to the cerebellum, as well as to the thalamus, and to other parts of the brain that need to be aware of our fields of vision. This is all to coordinate eye movements as well as balance cues. Here you can see that pathway of light. Notice the crossover occurring. You have your right eye and your left eye, and parts of those cranial nerves cross paths. So when you get that image to the back of the brain, this right side of the brain is actually receiving part of the left and part of the right, and this left side of the brain is recording or receiving part of the left and part of the right, which is why if you close your left eye, you kind of see bits and pieces of the room. It's not a complete picture, not just because one eye is closed, but also your brain is basically filling in the spaces of the things that you can't see, but your brain knows are there. Your optic nerves cross at a location called the optic chiasm that's right in front of the hypothalamus, and they follow something called the optic tract. Again, I said they're relayed through the thalamus, as all of our sensory information is. Let's move on to the ear. The ear has three main parts: the outer ear, which is receiving and directing sound waves into the ear canal, and it also contains spaces filled with air. The middle ear, which is conveying sounds and vibrations to the inner ear. It's within the temporal bone, and it has spaces filled with air as well. It contains the eardrum, which is also called the tympanic membrane, as well as the auditory ossicles, or the small bones that vibrate to conduct the sound into the inner ear. Our inner ear has the receptors for hearing as well as balance. This is also within the temporal bone. It's filled with fluid. That fluid is going to translate the sound vibrations into pressure waves that move through channels in an organ called the cochlea to stimulate the auditory receptors. There's also a vestibular apparatus, which is made of semicircular canals, utricles, and saccules. These structures sense position and movements of the head. Here you can see some of the structures I was referring to. Our outer ear, which is also called the auricle or pinna, directs the sound waves into that external auditory canal. Our external auditory canal has hairs as well as ceruminous glands secreting cerumen or earwax. It leads to the tympanic membrane or eardrum. Attached to the eardrum on the middle ear side is the malleus, one of our inner ear ossicles. Then we have the incus, and then finally the stapes. The stapes corresponds with our inner ear at a location called the oval window, which is not labeled on this diagram. It's corresponding with a structure called the vestibule. The vestibule contains two sacs, the saccule and utricle. Off of the saccule and utricle are the semicircular canals, as well as the cochlea. The vestibular nerve is attached to the vestibule. The cochlear nerve is attached to the cochlea, and these two fuse to form cranial nerve 8, which is the vestibular cochlear nerve. Another structure in this area is called the Gustation tube or the auditory tube. This is a structure that connects the ear and the throat or pharynx. This is to help equalize the air pressure behind the eardrum. This is why you hold your breath and push the air in the back of your throat when you're trying to pop your ears, so to speak. So how is sound perceived? The outer ear directs the incoming sound waves to the inner ear canal, then to the eardrum, which transduces the sound into vibrations. Those vibrations cause those inner ear ossicles, the malleus, the incus, and the stapes, to move and vibrate at the oval window of the inner ear. Those vibrations then set up those pressure waves within the cochlear fluid to move special hair cells within an organ called the organ of Corti. Action potentials are then elicited by those special sensory neurons of hair cells. The hair cells are those special receptors for hearing. Nerve impulses travel back through cranial nerve 8, the vestibular cochlear nerve, to the auditory cortex, which is located within the temporal lobe of the cerebrum. Here you can see what I was describing. Here's that, this is calling it the round window. It's actually the oval window in this case. It's a mislabel there. On the oval window, this would be where the stapes interacts, and the stapes is going to cause vibrations within this fluid, and that's going to cause these pressure waves to flow through the cochlea, and depending on the pitch, meaning if it's a high pitch or a low pitch, a low sound or a high sound, that's going to dictate exactly where in this cochlea that sound is absorbed, and that's how you get the determination of various pitches in sound. Here you can see that organ of Corti, as I was saying. So here's the actual temporal bone itself. This is a cross section of the cochlea. So this is a cross section of that snail-shaped organ that we were just looking at. Inside that snail, whoops, sorry about that. Inside that snail-shaped structure is that organ of Corti, which is attached to, or which has, I should say, what's called a basilar membrane or a tectorial membrane that has these little cilia on it. These cilia are going to vibrate when this fluid moves. When that fluid moves at a particular ratio or particular rate, it's going to stimulate those special hair cells. Those special hair cells will then communicate to this cochlear nerve that sound has been perceived. Here you can see that structure a little better. That basilar membrane or tectorial membrane with all of those special hair cells make up this structure called the organ of Corti. These special hair cells have lots of cilia on their ends, and that cilia is detecting movements within this endolymph or within this fluid that surrounds the structure. So as waves come down through this fluid, it's going to bend the hair or cilia on these special cells, and these special cells will then conduct an impulse down their sensory path. So how is sound perceived? Specific regions of that basilar membrane are going to vibrate with a particular pitch, as I mentioned. Higher frequency sounds are going to vibrate closer to the oval window. Lower frequency sounds are going to vibrate near the tip of the cochlea, or that inner part of the snail shell, so to speak. Louder sounds cause larger vibrations and higher frequency nerve impulses to the brain. Here you can see what I was mentioning. The sound comes in, beats against that auditory or tympanic membrane, which rattles these three inner ear bones, which knocks on that oval window, increasing the pressure within the perilymph. That perilymph is going to move and sway, moving the cilia on those hair cells. Those hair cells will then stimulate the cochlear nerve to communicate to the brain. Let's talk about the final sense, which is equilibrium. Static equilibrium is the idea of maintaining body's position, specifically the head, in relation to gravity. The saccule and utricle of the vestibule are involved in equilibrium. There are special sites called maculae that contain hair cells, not the same type of hair cells that we saw in the organ of Corti. They're covered by a crystal and gelatinous layer. Hair cells can be bent, and then the action potentials are generated to relay information about body position. Really, it's about head position, though, because those hair cells are not going to move if your foot moves, for example, but those hair cells will move if you decide to lay down or if you turn your head from left to right. Think of those hair cells in your ear moving back and forth as that movement occurs. So dynamic equilibrium is the idea of maintaining head and body positions when the body is in motion. These action potentials are generated to convey movement information. So whether or not you're moving, whether or not you stop moving, whether or not you're increasing speed, whether or not you're decreasing speed, etc. The ampullae detect this. These are found at the base of each semicircular canal. There's a special crista inside each ampulla that has embedded sensory hair cells. With movement, the change of position is going to cause the fluid within those semicircular canals to move and bend those hair cells within the crista ampullaris structure. So semicircular canals are orientated at right angles in the 3 dimensions of space to sense motion in all 3 dimensions. So up and down, left and right, back and forth. Nerve impulses can then be sent through the vestibular branch of the vestibular cochlear nerve to the medulla, the pons, the cerebellum, and the vestibular area of the parietal lobe. The vestibular nuclei, which is in the pons and medulla oblongata, receive information, integrate it with the information from the eyes as well as proprioceptors to coordinate muscle movements. Here you can see the anatomy of our static equilibrium device. So there's another layer of supporting cells, kind of like that basal membrane that we had before. You see these special hair cells. Hair cells are projecting their cilia into a special layer of dense carbonate crystals called otoliths. These otoliths are going to roll back and forth, or side to side, or up and down, or whichever direction as your head moves. And this is exactly what I was just describing. So here you see your head's upright, so those otoliths are sitting right on top of those little cells. But as you move your head, that's exactly what happens to your otoliths. They move, and as those otoliths, which is kind of like a jelly-like crystal, as they kind of roll to the side, that's going to pull those hair cells down, and that's going to send an impulse down the vestibular cochlear nerve. With dynamic equilibrium, so the equilibrium that's associated with acceleration or deceleration as well as rotation, you have the crista ampullaris structure, which is at the end of all of our semicircular canals. This is a cup-like structure which is going to move and bend as the fluid within the semicircular canals moves and bends. So here you can see the example. It's kind of the same idea as the otoliths with our static equilibrium. So when our head is in a still position, the cupula is sitting still, there's no movement. As our head begins to rotate, the fluid within our semicircular canals move. As that fluid moves, it's going to move that endolymph in different directions to move that crista ampullaris structure, which stimulates the hair cells to conduct an action potential. This animation goes through all of our special senses. And that concludes chapter